So it's a great pleasure. We have one of our new recruits to UCLA, a new faculty member in human genetics, Michael Wells, and he's going to be telling us about his very exciting work. Please welcome Michael.
Hi, everybody. Thank you for coming. My name is Michael Wells. I'm from Columbus, Ohio. And today we're going to talk about my exploration of human genetic and phenotypic diversity through cell villages. Let's get started. Okay. Our species is characterized by an immense diversity in neurological and psychological traits. We think differently, we behave differently, we interact with our environment in different ways. Now, while the origins of these differences have been debated for millennia, we're starting to now more fully appreciate that for many of these traits and the diseases associated with these traits, there's a certain level of innateness to them, meaning we're kind of just born this way. Now, this idea has been supported by decades of research, including twin studies that have been able to calculate high heritability scores for things like autism, schizophrenia, as well as personality traits like neuroticism and aggression.
It's going to turn up the volume.
Everybody can hear me? Good. Okay. More recently, genome-wide association studies have been able to pinpoint a specific genomic loci that are associated with traits and disease risk. And even more recently, large-scale exome sequencing studies have been able to nominate candidate genes as being risk factors for various diseases. Now, while we're doing a very good job of identifying the risk factors, the genetic risk factors associated with these diseases, we are lagging far behind in our basic understanding of the underlying biology. We don't know, for example, how these genetics influence some of the earliest stages of brain development, say how neuroprogenitor cells proliferate and differentiate, or say how postmitotic neurons form connections with each other and wire the brain. Now, for us to truly and fully understand these connections, we need to cut across many different levels of biology. We need to know how these genetic signals influence gene expression and signaling pathways and cellular phenotypes. We need to know how these phenotypes then in turn influence the ways in which the brain is wired and how this circuitry can ultimately impact behavioral output. Now, early in my career, as a PhD student in Guoping Feng's laboratory at Duke and MIT, I focused mainly on the right side of that schematic and trying to understand how genes influence circuits and behavior. And I did so using the mouse model. I studied certain genes like SHANK3 and PASH-D1, which have previously been shown to be involved as risk factors for various neurodevelopmental disorders. Now, it was during this time that even though I was able to recognize the immense importance of the mouse model, I started to realize there are certain limitations of this system. For example, it can be quite slow and laborious, which means it's going to be very difficult for us to keep pace with genetic discoveries in humans. In addition, no model fully recapitulates the immense complexity of the human genome. And as such, we need human tools that will enable high throughput investigations into human cells. And that's why, as a postdoc, I joined Kevin Egan's group at Harvard and the Broad Institute to study these disorders and these mechanisms using human stem cell-derived models of neural cells. Now, for those of you unaware of how this works, essentially we take human pluripotent stem cells in a dish, and we coerce them to become the many different cell types that populate the early brain, including neuroprogenitors, postmitotic neurons, and astrocytes. So one of the main questions I asked myself when I began my postdoctoral work was, can we use these human models for high throughput investigations into the relationships between genetic variation and disease-relevant neurodevelopmental phenotypes? And with that, I'd like to introduce to you our brand new experimental platform that I co-developed with members of the Egan Lab as well as Steve McCarroll's group at Harvard Medical School that we're calling a cell village. Now, a cell village is a system that allows us to connect alleles, molecules, and phenotypes. And this works by starting off with dozens, if not hundreds of different human donor cell lines that we culture all in the same dish. And in doing so, we're able to capture genetic, molecular, and phenotypic heterogeneity in a shared in vitro environment. Now, for the rest of this conversation today, I'll tell you not only how we do this, but also how we're able to extract biological information from this system. So, as an overview of what we'll discuss today, for part one, we'll talk about the village, how I create these villages, and how we can deconvolve these mixed donor cultures to answer important biological questions. Next, I'll give you a little vignette, a little story about how you use this system in order to understand the role of genetic variation in differential human responses to the Zika virus. Finally, I'll close out with a glimpse into the future, in which I talk about the mission of the Wells Lab, as well as how we plan on using cell villages in the future. I should say right up front, this work was funded by K99R00 by the NIH, as well as a Burroughs Wellcome Fund postdoctoral enrichment program award. Okay, let's get started. Talking now about the village, how we construct these things and how we can actually extract information from them. So this is the situation we are presented with. We really want to understand how genetic variation influences phenotypic variation in human cells. Now, the problem with that is conventional methods would require something like this. You'd have dozens of different plates with dozens of different cell lines cultured separately from each other. Now, the problem with this is it can introduce a considerable amount of well-to-well and run-to-run variation that can often mask important biological signals. In addition, this introduces a considerable number of challenges in control and rigor when you're trying to process numerous samples at the same time. Imagine trying to make RNA sequencing libraries from 50 cell lines from 50 different wells. It can get a bit messy, and you can have a lot of technical variation that gets introduced into your system. Also, this can be very costly, and it can limit the number of hypotheses that you can test at any given moment. So we asked ourselves, can we overcome all of these challenges by doing this, by putting all those different cell lines into a single dish? And that's what we're calling our cell village. So how does this system work? Well, as I mentioned, we start off with dozens to hundreds of different human donor cell lines. These are typically human embryonic stem cells or human-induced pluripotent stem cells. Now, for each of these donors, we have to perform either whole genome sequencing or SNP array profiling, because we need that genotypic information for later on in this process, as I'll describe to you in a bit. I then take each of these cell lines, I culture them separately, and I coerce them to become the many different cell types I'm interested in. In this case, we're going to focus in on neuroprogenitor cells, which I generated using what I'm calling the SNAP induction protocol that I don't have time to talk about today. What I will tell you is that it's a process that only takes 2 days to make neuroprogenitors and has worked in over 97% of the cell lines we've tested. Then once I'm ready, once I have the cell types I'm interested in, I can then put them all into the same dish. And again, in doing so, we're capturing genetic, molecular, and phenotypic heterogeneity in this shared in vitro environment. So you're probably wondering, well, if they're all together, how do we know who is who? How do we actually get information from this system? Well, there are 2 bioinformatic pipelines we use that we developed again in conjunction with the Carroll Lab. One is called Dropulation, and it's based on single-cell RNA sequencing of the cells in our village. Now, what Dropulation allows us to do is not only capture and quantify the transcriptional profile of each cell, but also re-identify the donor of origin for each of these cells. I'll tell you a little bit more in a few slides about how we can actually use this to answer important questions. We also have something called census sequencing, which is based on low coverage whole genome sequencing of the cells in our village. Now, this is doing exactly what its name implies. It's running a census on the cells in the village. It's asking who's in the village and what percentage of the village they represent. So for example, if I had 100 different cell lines and I did a really good job of counting them accurately and threw them all into the dish at equal contributions, well, census sequencing should tell me that each donor is 1% of that village. And as I'll explain in a few slides, I'll show you how we use this to actually capture phenotypic information from our village. Okay, let's talk a little bit more about how each of these works. Let's start off with Dropulation, which again is based on single-cell RNA sequencing of the cells in our village. Dropulation is an algorithm that leverages natural genetic variation across the donors in our village in the form of thousands of transcribed SNPs, and it uses this information to re-identify the donor of origin for each sequence cell. Essentially, we're using this information as an intrinsic barcode. So as you can see in this demonstration, what the algorithm is doing is scanning the genotypes at hundreds of variable genomic sites and calculating likelihood scores for each candidate donor. Now, eventually, one donor stands out above the rest in red, and that is deemed to be the most likely donor of origin for that sequence cell. You might also notice there's a second and third place finisher in this little race here. Well, those donors are actually siblings of the donor in red, showing that this algorithm is actually sensitive enough to discriminate between individuals that are related to each other. Keep in mind, though, the algorithm is using the genotype, the intrinsic genome, as the barcode, and so cannot discriminate between identical twins or say cell lines that are clones of each other. So it's one limitation of this system. So what can we do with dropulation now that we have this up and running? Well, we have the whole genome sequencing or SNP array data from each donor. We now have full transcriptional profiles for each sequence cell, and we can identify the donor of origin for each of our cells. And as such, we can perform expression quantitative trait loci or EQTL detection using this system. Now, if you don't know what EQTL is, it's actually quite simple. Essentially, you're finding associations between genomic sites and the expression of nearby genes. So for example, in this theoretical set here, the donors that have the AA genotype at this site have low expression of this theoretical nearby gene. And the donors with the CC genotype have a much higher expression level of this theoretical gene. So we can actually find these relationships on a genome-wide scale in a high throughput manner using dropulation. What else can we do? Let's talk more about census sequencing. So with census sequencing, it's quite similar to dropulation in the sense that we're using natural genetic variation as an intrinsic barcode. But in this case, what we're doing is we are isolating genomic DNA from these cells and performing low coverage whole genome sequencing. And we're doing this in an attempt to estimate each donor's contribution to that DNA mixture. So we get millions of sequence reads, and we're actually sampling each donor's genomes at proportions that reflect their contribution to the village. Now, of these millions of sequence reads, a decent chunk of them contain sites that we know to be highly variable across the donors. And these can actually serve as allelically informative SNPs, as you can see on the far left of this graph. At each of these sites, we can actually calculate an observed allele frequency for the village. So a village observed allele frequency, again, as you see. And all the set sequencing algorithm is trying to do is find a set of donor mixing coefficients that maximize the likelihood of those observations. So as you can see, this demonstration, what it's doing is it begins with the assumption that each donor is equally contributing to this mixture. So we have equal donor mixing coefficients. It calculates a likelihood score, makes an adjustment, and keeps doing this in an iterative fashion until those likelihood scores reach asymptotic values. This typically takes anywhere from 10 to 30 iterations for this to run. So you're probably wondering, okay, it's cool that you can figure out, the pie chart basically for your village, but how can you actually get information from this? Well, there's many ways to derive phenotypes from this data, but one way we're going to focus on today involves fact sorting. Essentially, what we can do is we can fact sort the cells from our village based on the expression of a marker of interest. You can then take your, say, high and low fractions, run census sequencing, and then calculate an abundance ratio from this, or the donor's relative representation in the two villages. This can actually serve as your quantitative phenotype. So we're going to talk about, in the next section, is a quick story about how I used villages, dropulation, census sequencing, as well as whole genome CRISPR-Cas9 screening to answer an important question about how human genetic variation influences our response to a certain virus. Again, this is work done with members of the Egan Lab and the McCarroll Lab, as well as some key players from the Broad Institute's Genomic Perturbation Platform. The virus we're talking about today is, for once, not COVID-19. It's actually the Zika virus. Now, when I made this slide and said that the Zika virus is an emerging global threat, that seemed real, and now I think most people don't think about Zika virus because it's nothing compared to what we're experiencing right now. Nevertheless, it's still an emerging global threat, and the Zika virus is actually a member of the flavivirus family. So these include dengue, yellow fever, and West Nile, and these are primarily transmitted by mosquitoes. This virus is characterized by periodic outbreaks, the most famous of which took place in late 2015, early 2016, in northern Brazil that spread throughout Central America and in the southern portions of the United States. Now, a vast majority of people who are infected with Zika don't even know. About 80% are asymptomatic. The 20% that do show symptoms tend to show mild things like conjunctivitis and fever and rash. However, there is a certain portion of the population that we're very concerned about, and that are pregnant women. Because if you are a pregnant woman and you're exposed to this virus, you are 17 times more likely to give birth to a child with a severe birth defect, the most prominent or famous of which is microcephaly, or small brain. Subsequently, other reports have found that infants that were, fetuses that were exposed to this virus in utero, that were then born, did not present with any sort of overt symptoms, were still at much higher rates, much higher likelihood of developing neurodevelopmental disorders later in life. Some of the earliest work in trying to study this virus found that it has a tropism, a preference for proliferating progenitor cells in the developing brain. And so it's with this information that we set out to try to understand the mechanisms by which this virus enters these cells and how our genomic variation can actually influence that susceptibility. So one of the first experiments I performed was I cultured 24 different progenitor cell lines that again I made from stem cells that converted using the SNAP protocol. And in separate wells, this is not a village, in separate wells, I infected these cells with either mock media or Zika virus. And I quantified the percentage of cells that were actually infected with Zika. And as you can see, some cell lines were completely resistant to infection, whereas others were showing nearly 100% infectivity rates. So the question I then asked was, is there any sort of human genetic variation that could explain this phenotypic variation that we're observing through this experiment? And so I'm cutting some things due to time constraints, but what I will tell you is I performed two major experiments in parallel to get to an answer. I began by creating a village of 44 different neuroprogenitor cell lines that were derived from human embryonic stem cells and performed dropulation-based EQTL detection. And in doing so, I identified over 960 E genes. An E gene just means it's a gene in which we found an EQTL associated with it. At the same time, I performed a whole genome CRISPR-Cas9 screen in a single neuroprogenitor cell line and infected those cells with Zika virus and asked the question, which genes either enhance or deplete infection? In doing so, we're identifying the genes that are important for the Zika virus life cycle. And the most important step here is I cross-reference these two gene sets. So I'm not only asking which genes are differentially regulated, but also which genes are important for Zika virus infection. In doing so, we limit this focus down to 8 genes. Of these 8 genes, I filtered by the effect size of the EQTL. So which genomic site had the biggest influence on the expression of the nearby gene? There's a very, very clear winner from this subsetting. We identified this SNP located on chromosome 11 denoted by the SNP identifier RS344-81144. As you can see, donors that have the CT genotype at this site have a very high expression of the gene IFIDM3 relative to donors that have the TT genotype. Now, IFIDM3 is a gene that we know to play a very important antiviral role in the cell. It plays an important role in innate immunity. And so based on these initial findings, we thought, well, perhaps the donors with the CC genotype, because they have greater expression of this antiviral gene, perhaps they are the ones that are most resistant to infection compared to the TT. And before we went any further, we actually looked into the literature and found some more data to support this assertion. First of all, it was shown that the presence of the T allele at this site actually enhances the repressive factors at the IFIDM3 core promoter. This is why we think these individuals have lower expression of this gene. In addition, another group actually figured out the mechanism by which IFIDM3 protects cells from infection. And what they found was that IFIDM3's protein product actually sits in the endosome of these human cells, in the membrane of the endosome, I should say. Now what happens is it can detect the presence of certain enveloped RNA viruses. And when it detects the presence of those RNA viruses, it can shuttle the endosome to the lysosome for degradation, thus preventing infection. Now, if you're missing this system, or there's a lack of this sentinel system, this increases the risk that these infected endosomes can actually uncoat and release the viral particles, where they will then form replication factories in the ER. So our hypothesis was then that the donors with the C genotype at this site are more likely to be on the left side of the schematic, more likely to be protected, compared to the donors on the T with the T allele at this site. So how did we test this? We went back to our village. We took our village of 44 different neuroprogenitor cell lines from 44 different individuals. We then infected with either Zika virus or mock media. And then 2 days later, we fact sorted based on expression of the Zika virus envelope protein. We took both of these fractions and performed census sequencing on them in order to get these little pinwheels. And then we were able to calculate the representation in the two. As you can see, our hypothesis was supported. The donors with the CC genotype had negative abundance ratios, meaning they had a greater representation in the Zika negative fraction compared to the Zika positive fraction. At the same time, the donors with the TT genotype had positive abundance ratios, meaning greater representation in the Zika positive fraction compared to the Zika negative fraction. Now, it was at this point that we then asked ourselves, well, perhaps this is just a byproduct of the village setup. By having them all together, maybe that's why we're getting this result. We wanted to know if it was actually intrinsic to each of these cells. So I went back to that original data set that I talked to you about a few slides ago, in which 24 cell lines were cultured separately and infected and quantified. Now, when I did this, all I did was I grouped this data based on the genotype at this site. And as you can see, it's pretty clear the donors with the CC genotype were the ones that had the lower expression, or lower infectivity levels compared to the donors with the TT genotype. I then created a replication data set of 36 cell lines that were not used in any of these previous experiments. And again, this relationship held true. And in this case, we were able to show that the genotype at this site could explain almost 60% of the variation in infectivity from this data. So it's typically at this stage of the presentation that people start wondering to themselves, well, I wonder what my genotype is. I can't answer that for you. I don't have access to your personal data. What I can do is give you some hints based on the 1,000 Genomes Project, in which they looked at allele frequencies across many different ancestral populations. And what they found was that ancestral populations in Japan, Southeast Asia, and China have very, very low risk allele frequencies. Okay? In some cases, it's completely gone. It's not non-existent. Same could be said about populations in Africa, and to a certain degree, South America and Central America. Now, this is a very, very different story, though, in European populations, in which the risk allele frequency was dramatically higher. Now, some of you may notice that the map I'm showing you here is color-coded blue and red. Well, that's because the red area corresponds to the terrain inhabited by the A. aegypti mosquito, which is the primary vector for Zika, as well as dengue, yellow fever, and West Nile. So this leads one to speculate that perhaps over the course of human history, there was some negative selection against the presence of this risk allele, driven by the presence of these viruses and these vectors. This is food for thought. This is something that I'm still thinking about. I know Noah Zaitlin and I have been talking about this a bit, about what this might mean. But to be clear, this is not just academic. There might actually be clinical applications and clinical importance to these findings. Because we know, at least it's predicted, that over the next 30 to 40 years, these viral vectors are expected to move further north into the United States and into portions of Europe. This is primarily being driven by climate change as well as increased globalization. So should this happen, in theory, we could have situations in which these populations are being exposed to this virus for the first time without having this sort of innate protection. However, on the flip side, perhaps we could use IFIDM3 expression as a way of mitigating this risk, or we could even start sequencing individuals during an outbreak at the genomic site we talked about today to identify those who might be most at risk for infection during an outbreak. So with that, I just want to give a quick summary of what I showed you so far today. Cell villages are a brand new experimental platform that allows us to capture genetic, molecular, and phenotypic heterogeneity in a shared in vitro environment. This is empowered by two bioinformatic tools, one called Dropulation, which allows us to do EQTL detection of villages, as well as census sequencing, which allows us to quantify phenotypic variation in our village. And as I just showed you, using this system, I was able to identify a single nucleotide polymorphism in the IFIDM3 promoter that plays an important role in differential susceptibility to the Zika virus. Now, I think that this is just the beginning. I think UCLA agrees, or else they wouldn't have hired me if they weren't confident this is actually going to work in the future. So let's talk about that future. Let's talk about what we're going to do in the Wells lab and what I think is the future of these cell villages. In theory, the cell village system can be used to answer a plethora of different biological questions that may be of interest to you, some of you in this room. We can create villages that consist of donors from any population. We can create villages of any cell type, whether it be in vitro or even primary human cells or primary mouse cells, whatever you're interested in. And we don't have to study these things just under static conditions where they're just sitting there in a dish. We can actually introduce village challenges, whether these be viruses or stimulants or patterning factors, or signaling pathway agonists, we can ask questions about how genetic variation influences the cellular response to some of these major signaling pathway agonists. In theory, we can also perform drug screens in which we ask how genetic variation influences the cell's response to certain approved or candidate drugs. I actually challenge all of you here to think to yourself, how can you use a village to answer some of the questions that are important to you? And if you have something really juicy, come find me. We'll talk more about how we can potentially pull that off. Let me tell you about some of the big picture questions I hope to address in my lab using not just villages, also non-village setups, three-dimensional cerebral organoids, various different projects or methods we plan on using in this lab. So first of all, I want to know, what are the molecular and cellular mechanisms underlying neurodevelopmental disorders like autism and intellectual disability? Essentially, what I want to do is use the tools that I developed as a postdoc to study some of the diseases I focused on as a graduate student. Next, I want to know if there's shared biology at the cell and molecular levels across the different disease genetic states associated with these diseases. This is a key that many people in this field are trying to unlock to try to identify mechanisms that might be shared across these different neurodevelopmental disorders. And finally, at some point in the near future of this laboratory, I want to start getting back into some of the model systems I used as a grad student, I should say, to understand the effects of these dysfunctional cell and molecular mechanisms on circuit formation and mammalian behavior. Now, for us to pull this off, we're going to need some people that are not interested just in advancing these cell villages to the next stage on the wet lab side, but perhaps more importantly on the dry lab side. So we're generating some really complex data sets that are starting to get beyond my scope of understanding. And so we really need some people who are passionate about addressing some of these questions. It's the final slide I'm going to show. Just to get into some more specifics about what we'll be doing in the Wells Lab. The first project we'll be focusing on is an investigation of the 16p11.2 microdeletion and duplication syndrome. Now, this is a project that's funded by an R00 mechanism for the next 3 years, and it focuses on this disease that involves a heterozygous de novo loss or duplication of 29 genes in this segment. It is one of the most common causes of autism. It accounts for approximately 1% of all cases of autism. And what I've done so far is I've generated a village of a couple dozen neurotypical controls and a couple dozen 16p patient lines in the form of neuroprogenitors. And I've generated many data sets, not just looking at how these cells interact or how these cells behave when static, but also in response to different powering factors and different signaling pathway agonists. Again, we've generated about 8 very, very large data sets that are just ripe for the picking. And I think there's a lot of really interesting data here that we can start to tease apart. So if you're interested in either collaborating or rotating or whatever it may be, maybe you just want to talk, whatever it may be, you can contact me here at my email address. In addition, you can follow me on Twitter, both my personal account and the Wells Lab account. So with that, I will open the floor for any questions.